here at Ectrums, we heard about vitamin D. Risk factors related to genetics and bacteria in the gut also suggest a fascinating connection to the immune system. Today, we'll hear more.
In terms of MS, everybody is an individual. They have their own unique course, a life course with MS. But at the same time, there are similarities. Take three groups of MS patients. One which was untreated, one was treated with Copaxone, glutamate acetate, and the third with any one of the interferon betas. So we extracted from the blood a molecule called RNA, which is basically the gene product. So genes make RNA, which make proteins, and then those exert functions on cells. Using those RNA data, we actually asked the computer to tell us how many subsets of MS patients were there in our data. Well, there were actually two groups of people that could be distinguished from a molecular point of view. But is it clinically meaningful? Does it really tell us anything about what the patients are experiencing? And so we looked at whether the subjects had relapses after the time they were profiled. One of the two groups, which we called MSA, actually had more relapses than the MSB group. And this is sort of an early observation that we are able to define two groups of patients, one of which may have a higher level of disease activity than the other from a molecular point of view. And I think what we discovered, this signature that defines two subsets, is just one of many signatures like this. And I think ultimately as we go forward, we'll have to integrate this with other signatures and gradually become more and more personalized in terms of the profile of the individual patient.
We're trying to develop a system that we're able to look at the effect of the gut and the bacteria in the gut on what happens in our brains. We know that if we take mice that have never seen bacteria or animals that have already seen bacteria, and we give them a certain strain of bacteria that is common to essentially all of mankind, that we can take those mice that would normally develop the experimental form of MS and we can protect them from developing MS by this bacterial strain. We can go even further and we can isolate from the surface of these bacteria a very specific molecule. This molecule, when administered to the mice orally, has the capacity to protect those mice both before we induce the experimental form of MS and after we induce the experimental form of MS. We're not mice, so what happens in humans, we know that if we take the peripheral blood cells from individuals who don't have disease as far as we know it, and stimulate them in culture, that we can drive their specific cells that affect disease, that cause disease, into cells that appear to regulate disease. So there's a switch from what's called an effector cell to a regulatory cell within 5 days. And the markers that are associated with those regulatory cells are markers that have been shown to be present but deficient functionally in patients with MS. We took blood from patients who have MS who had been off of any of the platform therapies for at least 30 to 60 days. And we took those peripheral blood cells, and again, this is preliminary, and what we found, interestingly enough, is that there was, first of all, a higher conversion rate to the regulatory phenotype in MS patients, and they had much greater production of IL-10 levels, which is very important in controlling the disease process, suggesting that MS patients, at least in culture, their cells appear to respond very favorably in the direction that is important and is critical as far as being a therapeutic agent down the road. This is really sort of the tip of an iceberg. It offers a whole new paradigm shift in treating MS.
That wraps up our coverage of Ectrums 2012. We hope that you've been inspired by the research developments that we've covered and that you have a better understanding of the group of individuals from all over the globe who are committed to creating a world free of MS.